Okay, I'm going to be covering quite a bit about viruses in general, not just B viruses. B viruses do appear, so don't get worried. They come towards the end, but B viruses aren't really as exciting as all the others. And they are very strange things. So what are they? And in particular, what impact do they have on living organisms? Note I say living organisms because there's a virus that infects just about everything. And of course, they are a cause of infectious diseases. But infectious diseases themselves have had the most amazing effects on both human and animal health. And they've also affected plants. And they've changed the history of mankind. That is really not an exaggeration. And they continue, as we know now, to have a very major effect. But I hope you'll see, maybe get our current situation in a little bit more perspective, because, oh boy, it could be worse. Hate to say that. So these are all nasty infectious diseases. Plague or bubonic plague and the Black Death, that was bacterial. There's been major pandemics from way back in the Roman times. In the Middle Ages alone, it's thought to have killed 25 million people just in Europe. That's really pretty bad. Typhus is caused by a very peculiar thing called rickettsia. They're a sort of bacteria. That's been known from the Middle Ages, and it affected the Napoleonic Wars and the Russian Revolution. And something like 2 to 3 million died in the First World War. That, again, these are astonishing numbers, they really are. And then, of course, when it comes to plants, we have the potato blight. That was a fungal disease, and it caused the Irish famine, which, as you probably know, killed millions and devastated Ireland. Yellow fever, that's a viral one. Now that affected Napoleon's attempts to invade Haiti. He went back. It's absolutely, most of his troops were ill. So a bit of a waste of time. Syphilis, that's another bacterial one, very ancient disease, extraordinarily unpleasant. And then we have tuberculosis and leprosy, which also are bacterial, still around now. Leprosy, infamous disease, and really disfiguring and unpleasant. Smallpox, another ancient and utterly awful disease. I've got a little section on smallpox, and that has been eradicated with a vaccine. So it's a success to have got rid of that. Malaria, that's caused by a protozoan. Well, a vaccine's been under development for so long, and still millions are dying every year. Sadly, this pandemic has held back the development of the vaccine, and the death rate from malaria has gone up a lot because, of course, it's in poorer parts of the world where they're not coping with this pandemic. Then some of the real nasties is this, whoops, sorry, go back. To these hemorrhagic diseases, Ebola, Lassa, whole load of them, they're viral, incredibly contagious, and have a very high death rate, much, much higher than anything else we've seen. The big worry is that they could become pandemic because of the way people travel. Now, there is a vaccine under development, and that was the fastest vaccine ever being developed. And we've just got to hope that that continues in development, because most of the time we don't hear about it because We're rather self-centered in the West, and after all, it is in one of these countries we don't know much about, but they are horrible diseases. Measles is viral. Now that is controlled by a vaccine, but it's still a major killer. And one of the reasons for that, unfortunately, is I think we have to put down to this chap who tried to link it, the MMR vaccine, with autism. And there is a real reluctance, particularly in America, for people to be vaccinated. The measles can be controlled, but you've got to have, I think it's approximately 95% of children vaccinated before you get herd immunity. So I think with this latest pandemic, just forget herd immunity. Polio, another dreadful one, almost, I'm going to come to that, almost eliminated with vaccines. It's so sad when there are vaccines that work, and yet we still can't get rid of things. Foot and mouth, of course, is an animal disease, large animal disease, viral, widespread. You probably recall the awful outbreak we had about 15 years ago, I think. Influenza, another awful viral one. There was a major pandemic in 1918 to 19, just after the First World War. It affects animals, non-human animals as well. In fact, there's a nasty outbreak going around now of avian flu, quite worrying because it's a very unpleasant strain. It's not hit the news, although I happen to know somebody who works in the team on it. They are working flat out on it. And then, of course, we've got HIV/AIDS, viral. They have been trying to get a vaccine for that for decades now, and still absolutely nothing. So really bad. Now I'm going to look at a couple of the real big major viral ones. Quite scary, these. Poliomyelitis or polio. Now, if anyone around is as old as me, they'll remember polio. It was really frightening. It is actually thought to be a very ancient disease, and people have assumed that this is, you see, notice it's got a crutch and it got a withered leg, thought to be an ancient disease. But this may sound very contradictory. It's actually a disease of the 20th century. And strangely, it was accentuated by improved hygiene. And there was a vaccine developed initially by Salk in the US, and then subsequently Sabin, an oral vaccine. And I am old enough to remember when they rolled out this vaccine a really big way. And I can see them having to do the same sort of thing now. They concentrated on the young, and I had just started work temporarily. And I think all schools, colleges and firms, I don't know if it was mandatory. I didn't inquire. I was just told when I turned up for work, there was a coach, you got on the coach and you went to some local place and everybody was vaccinated. It was a huge breakthrough. This was the scene one saw on the news, polio patients. It affected the breathing, and these awful iron lungs just look rows and rows and rows, predominantly children, but not all children. And they are really the forerunner of the current ventilators that they're using now for coronavirus. And it terrified people, it really did. And Franklin D. Roosevelt, President of the United States, was a sufferer. He didn't get it till he was 39, so it wasn't all children. And this poor little girl has withered legs and has a sort of crutch thing on her leg. And this was part of a big campaign in the States. I think it was called a dime or something, and they collected huge sums of money for research on it. Roosevelt never liked to be seen in a wheelchair. You'll nearly always see pictures of him behind a desk, but he was polio victim. The great thing about this, of course, it was oral, and it was given as drops on a sugar lump. Now that's what we want now, not some wretched vaccine that's got to be kept at -80 centigrade, which is crazy. But that was a terrific breakthrough, it really was. They've been used worldwide and they have almost eradicated it. And very recently, this was a great moment, they eradicated in Africa. It's been eradicated in most of the world, but antagonism to vaccination has prevented it being eliminated. And this is the kind of thing that happens. A suicide bomber killed over 15 people at a polio eradication center. It's Pakistan and Afghanistan, so I don't need to tell you the politics, but it is so worrying that these people, they put about all sorts of stories of the Western world trying to do terrible things to people. So the people, many of the medical staff trying to give the vaccine are killed. The other big horrifying disease was smallpox. This is a really ancient one, and there's evidence of it right back into the 3rd century. And this is nasty because it spreads between both people and infected objects. And in 18th century Europe, just Europe, it's thought that 400,000 people died that year. The numbers with these diseases are really quite frightening. So it puts in perspective a little bit what we're hearing about now with the numbers of people dying and the numbers of people with COVID. 6 monarchs died during the period, and it's thought there was 300 million deaths in the 20th century and 500 million in its last 100 years. And 15 million in 1967. But this is a wonderful success story. One of the really big names in this field, Edward Jenner, he was called usually the father of vaccination. And variola, it's often called, this record's going right back in the 1500s to China inoculating people. Well, Jenner noticed that milkmaids often didn't get smallpox. And of course, cows are affected. And he associated this, good observation, with them perhaps getting mild cowpox from handling cows. Could this perhaps in some way be protecting them? Remember, at this stage, we don't know what virus is. We'd no idea what was causing it. We didn't even know much about bacteria. And if they handled cows, they just got a few pustules on their hands. And it was widely known in the country that this might be so. And a lot of people actually inoculated themselves. So there was some sort of control. So Jenner tested this in 1796. He did things that would never, ever be allowed now, but it was successful. This sad picture, that's just so sad. Look, this poor child. It's not just a few pustules, is it? It's hideous. And this is very famous. This is in the Wellcome collection, because this is the young girl who has cowpox. He took samples from these lesions. But the wonderful thing was on December the 9th, 1979, the Commission of Scientists declared that smallpox was eradicated. It actually carries around a 30% chance of death. That's only equaled by Ebola. It is awful. And it's the only infectious disease that's been officially eradicated. And how sad is that when we have it in our power to eradicate others? This picture, again, you will see time and again if you look up anything on smallpox. It was taken in 1901 in the Leicester Isolation Hospital. They're twin boys, and this lad had his vaccine. This chap missed the vaccination. And just look, it's an absolutely brilliant example of what vaccination can do. And then the WHO had this marvelous banner across their magazine, May 1980, smallpox is dead. Worrying thing, of course, is that the virus is still in the vaults in a couple of places. So if you want to write really nice thrillers, of course, that's what you can base them on. However, there were anti-vaxxers even then. This is a brilliant cartoon by James Gilray. You know, he was a wonderful cartoonist, 1802. Now, I don't know what big screens you've got, but if you look closely, it says at the bottom, the cowpox, or the wonderful effects of the new inoculation. And they're really dreadful. They've got pictures of people with cows coming out of their noses, cows coming out of his backside, cows coming out of his hands. They definitely weren't all terribly keen on this awful thing called inoculation. I think I've probably got a pause there in case anybody's got any questions relative to that bit of the talk. They might like to bring them up now.
There is a question. How did improved hygiene accentuate polio, or have I got that wrong?
No, you haven't got it wrong. I think probably it was assumed that when things were much dirtier, people picked up very, very low levels and there would have been some sort of immunity. But gradually, it started to spread as things came cleaner, and I think the background levels fell. It got to the stage in America where they wouldn't let their children out to go to swimming pools because it was thought it was spread probably by swimming pools. It's the one I can remember having. I used to get swollen glands, and I do remember my mother obviously being desperately worried, the doctor coming, and I heard, close the door, and I heard saying to my mother, "Don't worry, little mother, she has not got polio. It's just swollen glands." I think they had, I don't know, tightness around the neck or something was an early stage, but it was something that people were panicked about. And the assumption was that it had been pretty widespread, but most people had picked up very low levels. I believe I had a question sent in earlier. I don't know if that person's there, about can you develop resistance to vaccines like you can to antibiotics? And the answer to that is no, it's quite different. Either a vaccine works or it doesn't, but you cannot become resistant to it. It may not work in you for varying sorts of reasons, but becoming resistant, it's not you that becomes resistant, of course, it's the organism that becomes resistant to an antibiotic. And that's simply because they're terribly good at finding ways round the antibiotic. They'll block it getting into the cell, they'll export it, they'll devise an enzyme. But vaccines should work. They do work largely on a very wide range of people. There are exceptions, of course. It is said that older people don't put up such a good immune, react so well, but there isn't any clear evidence that there's any kind of resistance to it.
Right.
Were there any others?
Yes, there are. What's the difference between vaccination and inoculation?
Well, it's just inoculation early on was what he was doing. It is what we would now call vaccinations, because you are inoculating with either with that, it would have been a weaker form of the disease. a lot of vaccines still being used now are what are called attenuated. So they're a weakened form of the actual virus or bacteria. The newer ones are quite bizarre. Goodness knows whether they're going to work. But you are inoculating. So you're sort of, this is why people were worried about, in a way you are actually infecting people. If it's an attenuated vaccine, you are actually giving them a form of the virus or bacteria that's too weak to produce disease, but is sufficiently like the disease to trigger an immune system, your immune system, to get going and recognize it as something it should be fighting.
Right, this one follows on very closely to that. Is it right that we used to give a live form of the polio vaccine in the UK when this was not allowed in Europe?
I don't think so, but I can't be positive about that. I have a wonderful book called Viruses, Plagues and History by Michael Oldstone. And the most revealing thing about that, with what's going on currently with COVID vaccines, is it has always been the same. The infighting and the politics behind it are quite frightening. So I don't know, it might have been, there were differences between here and the States, and there were enormous arguments between the Sabin and the Salk. And there was another person, and I can't quite remember their name, but the infighting that went on was enormous. There was a group in Scandinavia that were pushing one of the kinds of the vaccines that America didn't like. But the politics that went on in the background was enormous. It really was.
There is another question, but I think it might be a bit controversial for a BBKA talk. And is that, would you persuade everyone to have the COVID vaccine?
Yes, I would. I'm very pro-vaccine. In spite of all the hassle that's going on, I don't believe that our authorities will allow something out there until they have done at least reasonable level of trials. It's very early days, and it is worrying that they're pushing them through so quickly. But they're not there yet. I mean, America are going overboard, as of course they always do, on the Moderna one. But the FDA have not allowed it yet because they're still saying they want to see more results. And unfortunately, you've just got to wait until you see the numbers of people who've been, they're all double blind. So that means the doctors and nurses don't know what they're giving, whether it's placebo or whether it's vaccine, and the people receiving them don't know. They have to be double blind because otherwise there's a lot of prejudice into what you get. The current two at the moment, I think what is upsetting is certainly the early one, the Moderna and AstraZeneca are putting forward all this information, a bit like Trump, in press releases instead of in scientific data. And that is really worrying. So they're estimating the 90% or whatever it is on a handful of people. They've dosed an awful lot of people, and then they wait to see how many people come up with symptoms. And they waited till they'd got, I think it was 95 people out of the thousands that had been injected. And they then unscrambled the results of those people, and the vast majority of them had received placebo, so no vaccine, and a handful, and I cannot remember, I didn't write down, I have a terrible memory, something like 5 or 6 or 7 people actually had been vaccinated. So the figure they produce is actually only on that small number of people. So I think until they have more, and they didn't say on the Moderna one, when they said they had symptoms, what kind of symptoms, how serious were they, where the other one is BioNTech and Pfizer, and they have given more detail. And I think there were just 1 or 2 had moderate symptoms and the others very mild. So that is very promising. But I think I'd still rather wait till there are a few more. But I think the regulatory authorities are not going to allow anything out there, certainly not in the UK, until they've got more. The Oxford vaccine is further behind, but they're coming through with good results as well. And then there's loads of others in the pipeline. I mean, they are going to come through, I think, probably thick and fast. But no, my own opinion is I've signed up for, I don't know what it was, something in the NHS appeared online, which I signed up to a while ago, saying that I would volunteer for one of the trials, and they want people of all age groups. So you get called or you don't. But yes, I would, definitely.
Right, there's one more before we let you move on to the next part of your talk. What is the main difference between bacterial and viral infection, and how do you find out for the first time when you get something, whether it's bacterial or viral?
What if you have an infection, you mean?
Yes.
The answer is they often don't know. If only they did. One of the big crunches, which is one of the things that affects the problems over there being a lack of new antibacterials, is there are hardly any bedside tests. They have to go by signs and symptoms. They collect samples in hospital if somebody's seriously ill, blood, urine, might be all sorts of things. And if it's a bacteria that they can grow quite easily, it will show up. But even when you've got a bacterial infection, the figures are really quite scary in that you don't always manage to culture it. So they're treating you not knowing really what you've got. And quite often, because fungal infections are much rarer, but they suffer from the same thing. So if they can't grow anything, and they might then say, well, we don't think it is bacterial, it's a question of signs and symptoms and then trying to dose. And of course, there are very, very few drugs for virus infections anyway, very few. HIV is the exception. There are a lot of drugs there and a very small number for some things. So it's a bit of a shot in the dark with a lot of them. But even streptococci, I mean, there's one or two. The problem is that there would be money in it, but nobody puts money into diagnostics. And that's what they want, is diagnostics. They won't be able to come to your bedside, collect a sample, put it in some sort of test kit and say, definitely that's pneumococcus or whatever. And then they can use the correct antibiotic. But unfortunately, a lot of the time they can't do that.
Okay, right. That's enough questions for now. We will let you go on to the next section of your talk. Thank you.
Right, where are we? Polio. Louis Pasteur, another of the great names. Everybody must have heard of Pasteur, pasteurization. He was the most amazing man. Pasteur's done just about everything. He put forward what was called the germ theory, and very handsome looking man, lived from 1822 to 1895. And now he proposed that it sounds obvious to us now, but that certain diseases are caused by the body being invaded by microorganisms. Remember, we've hardly got round to finding out about microorganisms. They're too small to be seen other than through a microscope. And the theory around before that was something awful called spontaneous generation, where, it was very obvious if you left a piece of meat somewhere, you saw nasty things growing and flies growing on it. So they thought spontaneous generation, but he was correct, of course. And he had a little go at everything. He worked on fermentation. Here he is looking on fermentation. Saccharomyces is the yeast that's used in both brewing and winemaking. It's a typical Saccharomyces budding, Saccharomyces cerevisiae. And he did these absolutely famous experiments. I do recommend this that I put down here, How the Scientific Method Works. it's a website called howstuffworks.com. It's just superb. He's lovely, very simplistic, but it sums up what he did. He had broth in each one of these flasks. He heated them so he would kill anything living in the matter. Now, with all these brilliant experiments, Mendel onwards, there's a little bit of serendipity, because the clever clogs will say, yeah, but supposing it was a heat-resistant organism. Well, fortunately, it wasn't. So he then sterilized, they were sterile, and he left them at room temperature. But of course, that's exposed to the air. So's that one, but it's got this lovely long swan neck, which stops the organisms getting in. And of course, you will know, obviously, what happened. That one became contaminated and that one didn't. In fact, they still had for decades, he had some other ones that he sealed off the end. And many, many decades later, they still remained sterile, which was extraordinary. And the word contagion was invented by this delightful man, Girolamo Frascatore. And he said that was an infection that passes from one thing to another, which is what we would say now. He also said that an epidemic disease caused by transferable tiny particles. Look how long ago this was. Long before anybody else had thought of this, tiny particles or spores that could transmit infection by direct or indirect contact. I think it's extraordinary. All the way through all kinds of science and biology, you find these people who are not just decades, but hundreds of years ahead without any evidence, but they work out something that is actually right. So that's contagion. But it isn't until we come to the microscopes that we really start to see movement. The early days, well, one of the earliest was Robert Hooke, 1635 onwards. Now, this is one of his microscopes. It is so beautiful if you look at it, exquisitely made, it really is. And this is in a museum, the micrograph. And this was designed by him and made by somebody called Christopher Koch in London about 1665. And Robert Hooke used it for his book called Micrographia, which documented a whole load of observations. Now, it had 3 lenses and it could magnify 50 times. We're going to move on to something much better, but that was thought to be fantastic at that time. It is, still is. And the first thing he noticed was slices of cork. And in 1665, using that microscope, he noticed that they were all composed of little boxes or cells. And the word cell, cella in Latin, means little room or cubicle. And of course, living things are all composed of cells. But that was the first observation of that. And this cell theory became very important. And this is rather nice because we've got both a botanist and a zoologist who often are on opposite poles. And they have agreed with this cell theory. We've seen that all organisms are composed of essentially like parts, namely of cells. All living organisms, in fact, no matter how complex they are, they do arise from a single cell. When a sperm meets an ovum, which is going to give rise to a human being, it starts off as one single cell. So everything starts as a single cell. And this is Rudolf Virchow. A little bit later on now. I do love his study. It's so delightfully tidy, isn't it? And he was an advocate of the cell theory and said that all cells come from cells. Recently, Professor Sir Paul Nurse, you might have heard him. He's been very voluble indeed about the muck-up that's been going on. He's just written a delightful book called What is Life that I can highly recommend. It's a lovely read. It's very simple and straightforward, beautifully written. And he says the cell is a fundamental structural unit of life. And he calls it biology's atom, which I think is a super description. He is a Nobel laureate, geneticist, biologist, superb man. But moving on with microscopes, we have this amazing chap called Antonin van Leeuwenhoek. I'm assured that's how you pronounce it, Leeuwenhoek, Dutch. Now, when it comes to looking at objects, we can only go down to about a millimetre, but we can't see detail. Below 0.1 definitely can't be seen by anybody with a naked eye. But this light microscopy allowed this whole world of microscopic life to be revealed. And we've already seen cork cells, but of course, when it comes to viruses as well, as we see, their small size hampered work hugely, because these are the people who are all assuming things. Van Leeuwenhoek, he made microscopes that could magnify up to 500-fold. I still find this extraordinary. Hooke, 50-fold. He's talking about 500-fold. I mean, a light microscope, it's got to be quite good. If you're going to look for nosema, you've got to do up to 200. But because of that, of course, he discovered a huge number of microbes and cell types. Whole world out there. It's a beautiful picture, I think, of him. He looks really rather lovely. Many years later, there are no lenses that could actually compare still with his. He wouldn't reveal his techniques, and it wasn't until the invention of the electron microscope that we could go down even smaller. Van Leeuwenhoek was famous for what he called his little animalcules. He took some scrapings from his teeth, and he prided himself on his mouth hygiene. And he was horrified to find all these things, some of which were swimming around. So as we move on, of course, with light microscope, we can see bacteria. Now, bacteria will grow up to give you visual colonies if you streak them on an agar plate, but they need a microscope to see them. There's a scale there of 10 microns, and these are cocci, staphylococci. But the real breakthrough comes, of course, with the electron microscope. And very strange thing, they started to use I think the very first work was back in the 30s, and work ground to a halt during the war, but then took off very, very much after the war. And as you move into the 50s, so electron microscopes started to be used. I used electron beams, it had to be in a vacuum, and this allowed visualization even of viruses. They make very thin sections, negatively stained, and they use heavy metals. And TEM is transmission electron micrograph, and they can go down as small as 20 nanometers. The other kind is called scanning EM, and this is an example of a scanning EM. They always color them because they think everything has to be pretty. But the joy of scanning EM is that it's 3D. So you can actually see that this is a staphylococcus. Transmission are sections, and to be honest, they're rather gloomy. They're nearly always greys, and they are so detailed that only the real expert can understand what they are. Now, we'll mention here, you notice these little tiny holes. These staphylococci are on a filter. We're going to come to filters in a moment. They've been filtered on something which has held back the staphylococci. because they're much too big to go through the filter, even though they're pretty small. Now, viruses are also microbes, but boy, are they strange. At least we classify them as microbes. They are ubiquitous, and I really mean that. They are just everywhere. There are more viruses than any other form of life on the planet. They can infect or inhabit anything, any plant, any animal, any microbe, wherever they look, they can find viruses. Now they know what to do. But they're really different from other microbes. They're way smaller. They're way under this 0.1 of a micron. So they're not visible with a light microscope, no matter how good the light microscope is. They don't metabolize, they don't respire, and they can't exist outside any other cell. This is an important point too. They can't move to find a host. A virus isn't going to leap at you from out of a tree. So they are odd, we'll be putting it mildly. Now, they have things called nucleic acids, which are normally called the genome. Now, the nucleic acids are either RNA, which stands for ribonucleic acid, or DNA, which is deoxyribonucleic acid. You can't fail to have heard of DNA. Now, in fact, the virus we're all so concerned about today is an RNA virus. But most organisms contain both of these. And the genome is required by all living, please note this word, living cells to reproduce. RNA is more flexible than DNA, but is much less stable. There are reasons for that. That can lead to problems because it means that it's prone to mutation. So the RNA viruses are much more prone to mutation than the DNA ones are. But so these nucleic acids have to be in everything to allow them to reproduce. Don't worry about this, but I know some people like to see this picture. The double helix, which is DNA, notice there's two strands in a helix, and the single one, which is ribonucleic acid. They all have what are called bases, base pairs, but you will notice for DNA, one of them is different. So you've got cytosine in both of them, guanine in both, adenine in both, but DNA has thymine, uracil is in RNA. Now, the viral properties, the things that make them very odd. For a start, they only have DNA or RNA. They never have both. And that's determined by the various sugars. Either they've got ribose, which is RNA, or deoxyribose, which is DNA. They have huge, I've got some pictures because they are bizarre. The shape, the size, and the symmetry of viral particles can vary really wildly. One of the good things about viruses is you don't have to worry about classifying them. I mean, there are virologists who like to classify them, but for most of us, it's just not important. Some viral particles may remain dormant. Fortunately, not many of the nasties. And there's absolutely no evidence that COVID does. So how do they differ from other microbes? Well, they're only one of these. These nucleic acids can't replicate outside a cell. The virus has a protein shell, often called a capsid, and it might have an envelope, which comes from membranes of the host cell. The famous COVID does. Now, the thing is, they can only reproduce inside some host cell, so not their own. How are they going to get in? They've got to attach to a host cell, and this capsid, this outer bit, has a protein which has a very high affinity to a host cell protein. So you've probably heard about the spike protein on corona, and it's that spike protein that latches on to our cell protein. And that allows the virus to get into the cell. Otherwise, it can't do anything at all. And then when it gets in, it takes over the cell and it tells the cell what to do. It just reorganizes the entire cell to make it produce new virus particles. And usually the host cell dies as the virus particles release. Once it's made enough, the virus particles then come out. So this is a very simplified, they call them infographics, I think, but they are rather fun. So here is a body cell with viruses. They enter the cell, being engulfed there. Then inside the cell, quite often the cell itself will strip off the outer coat of protein. It doesn't quite know what it's doing. And then the nucleic, oh, drat. So I keep pressing the mouse in the wrong way. And then the nucleic acid is released. Then, of course, the virus has got a marvelous time because all it's now going to do is to take over the cell's chemical manufacturing system. So the cell ignores its own needs, whatever else it was doing, it has to give up, and it just makes more and more viruses. And very often, of course, it destroys the cell. So they are thus, they're intracellular because they're growing inside a cell. They're obligate pathogens or parasites. Obligate means that's the only way they can exist. They're obligated to be that. The extracellular phase, the bit outside, is inert. It doesn't do anything. So COVID virus hanging about the place is perfectly okay. It can't do anything unless it can get into a cell. And the same with all of them, whether it's deformed wing virus, anything at all, can't do a thing. But when it's intracellular, it's then just a replicating machine, completely replicates. They are often very, very specific. A particular virus depends on its ability to attach to a host cell. This is a little bit like pollen grains when they land on a stile. Only the right pollen grain can do anything. If it's a foreign pollen grain, it's not recognized and it can't penetrate down. So it's got to be able to attach. And this was just like the pollen grain, there's got to be a receptor site on the host cell. And it's also got to have a specific adsorption site in the capsid or the envelope. So it needs quite a lot, really. Very rarely a virus can infect completely different hosts, but it is extremely rare. I found this one, haven't a clue what it is, I might say, not a norovirus, but it's a mosquito pathogen. And apparently it can cause paralysis in honeybees, but only when it's injected into them. I don't think it's ever been found in honeybees. So that's quite a big gap. And it can cause a serious disease in pigs. But that seems to be very, fortunately for us, I think, is rare. Now, just some pictures because they are weird and wonderful. This is a filamentous virus. This is the hideous Ebola, and that's a scanning EM of it. It just looks evil, doesn't it? Polio virus, which is a rather shapeless looking thing. And this is just a very brief outline of the kinds of sizes and shapes. Polio is one of the smallest, and smallpox one of the largest, and Ebola, that really weird long thing. And they're, roughly speaking, there are filamentous ones, there are hexagonal, icosahedral. These are ones that have 20 sides, very strange looking shapes, enveloped or complex. And they're sometimes classified by their size and shape. This has got to be the most peculiar one of all, and it's called a bacteriophage or phage. I mean, that obviously is a sort of diagrammatic one, but the pictures of them really are like that. It's got this large head and these strange things. It looks for all the world like a landing craft on the moon, I think. But there are they, a load of phage infecting a bacterial cell. And that's transmission electron microscopy. And you see, they do look like that. They're very, very odd. They were thought to be a jolly good way of controlling bacterial infections way back. But unfortunately, they are so specific that they're not much good to work on. Because, I mean, for example, it won't, a particular one, this is an E. coli, and it doesn't affect a salmonella. And one that goes for a staphylococcus, it's only a particular kind of staphylococcus. So there's not much future in that. So how do we actually find out about viruses, remembering that we had not got any idea before what they were? Well, Louis Pasteur had shown bacteria and fungi, and he knew that they could be removed by filtration. Remember, I showed you the picture of the staphylococci on a membrane filter. Initially, they had things called porcelain filters that were used. And if you passed a liquid through that, you would remove all the bacteria and fungi. So therefore, if anything went through, when it came out the other end, it should be sterile because you'd removed everything. And then became clear that that wasn't so. Now, the very first virus discovered goes quite a long way back, and it was tobacco mosaic virus. Tobacco, remember, was a huge crop, massive amount of money. And it causes great damage to the tobacco plant. And it's a good example of how it's never really one person, it's a collection of work. First of all, this chap Mayer showed that you could transfer it between plants. You could rub the leaves on one another, contact. Ivanovsky showed that it wasn't bacterial because it went through a very fine filter. And instead of being sterile at the end, if you put that liquid onto a healthy tobacco plant, it became infected. So it was filterable. And then that work was repeated by somebody else who replicated it. And he coined this term virus. And that was about in the 1890s. So it was called filterable. But this is really intriguing. Look, they crystallized it. And those are crystals of tobacco mosaic virus, which leads us to say, is this thing living? Then we have this chap in 1898 with a very modern looking microscope, and he worked more on it and again confirmed that it was filterable and contagious, and he used the word virus. Quite a bit later on, Wendell Stanley, an American, discovered that they were made of nucleic acid and protein only. Remember that we've said that they all had a nucleic acid and a protein, but nothing else. So questions have been asked time and again, are they actually alive? Why are we saying that? Well, most microbiologists think of them as being on the edge of life because they don't have a cell, they haven't got a nucleus, they haven't got a ribosome, they've got no cell wall, and you can actually crystallize them, which seems very odd. You can crystallize chemicals, but to be able to crystallize something that is living seems rather odd. And these virus particles or virions are completely inert outside a host cell. The vast majority can't retain the ability to infect for long. This is quite comforting. Just remember that one. And unlike a lot of bacteria and fungi, they cannot produce any long-lived viable, for example, spores. So they're very unlike other living things. So I have pause there before we move on to viruses in bees. Do we have any questions?
What are prions? Are they different from viruses and phages?
Oh, what a lovely question. Prions. That's the latest thing that life has to throw at us. Prions caused enormous controversy because they were thought very early on, CJD was the nasty thing. It had been known for a long time, and I forgot the name of the other one, in some island where they ate one another. And they were often called slow viruses because they knew that whatever it was that producing what appeared to be an infection took years. It's thought that some missionaries went out with CJD and they were eaten, and the prize when you eat humans apparently is the brain, and they ate the brain. So they were thought they definitely were in a group called slow viruses, but nobody could find any evidence of that. When they then started all the work, because of our stupidity of feeding sheep to a ruminant, because it's a good get-rich-quick thing, it was eventually discovered that they are mysterious misfolded proteins. And they were arguments galore, just like our virus is really alive. Couldn't understand how a misfolded protein could actually act like an infectious particle. But that's all they are, a misfolded protein. They haven't got anything else there at all. And there's still a lot they don't know about how they cause the effects they cause, but they are intriguing.
There is a question from Gareth. Can all viruses spread horizontally and vertically, or are they selective?
What are they? What?
Selective.
Selective. I think they can do either, but not all. No, it varies very much as to what the virus is and what the conditions are. And remember that there's still an awful lot not known about a lot of viral infections, even the ones that are being worked on. When it comes to bees, of course, it's even more difficult. There's so little known. But so I would guess that it is going to be variable.
Okay, I think that's the last question for that part of the talk. There is another one, but I think that will come at the end.
Right. Right, so on to bee viruses. These are quite a problem. The knowledge is way behind that of other animal viruses and the commercially important plant viruses, because there aren't many that cause a vast amount, or didn't used to be, cause any kind of devastation. Some have been known for a long time, but the understanding of them is actually pretty sparse. Sac brood virus certainly has been known a long time and was one of the first to be identified as a virus. It was reported right back in 1913. And then again, a little while later, same chap working in Denmark and Australia. And then in the 60s, it was confirmed as a virus. Because remember, until you've got the EMs, being available widely didn't happen until well through the 50s and into the 60s. I should have mentioned the early transmission electron micro scanning came later. I remember them at work, and they were vast, a huge, huge room with this enormous thing, a bit like pictures you see of early computers, you know, right down now to a little mobile phone. It used to take up a huge room. So they were desperately expensive. And it wasn't until you got well into the 60s that there was any use made of them. This is a familiar picture, isn't it, of the Chinese slipper. Strange little thing. Now, he showed, White was this person I mentioned a couple of slides ago, he showed that a filtrate could transfer it from larva to larva. And remember, if you've got a filtrate, you've got rid of your bacteria and fungi. So therefore, it's almost certainly going to be a virus. Interestingly too, that if he heated it, that lost its infectivity. So that all indicated that it was a viral disease. Bailey subsequently worked with Brenda Ball, and they are the big names in bee virology. Brenda Ball is still around. Bailey has died now, and his book is magnificent, it really is. And they studied the infectivity. Now, it's a hell of a lot of infection here. For those who are not familiar with these figures, 10 to the 6 is 1 million. So 10 to the 7 is 10 million, 10 to the 8, 100 million. So between 10 to the 7 and 8 particles were required to produce disease in a 2-day-old larva. So it's actually an awful lot. And when the larva was infected, the numbers went up to these astonishing levels. So of course you then say, well, how infective is that? This, I don't think, is particularly infective, but once you've got that number of particles, that's quite bad name, but quite bad news, it really is. He determined that it was hexagonal in shape, and that was done using negative staining TEM. So Rothamsted were well ahead of the game then. They'd got a TEM up and running. The size is 28 nanometers, so it's one of the smaller ones. And then other workers have showed that you can find the viral particles in the cytoplasm, in the fat and muscle of larvae, and in the fat bodies of adults. But in fact, of course, it doesn't actually cause a great deal of damage in a colony. It doesn't actually kill the larva until after it's been sealed, and it prevents it entering its final molt, prepupa to pupa. And you still get this tough cuticle, so that fills with molting fluid, which is what gives that great big sac. Well, of course, that is containing huge numbers of virus. And if the adults try to remove the diseased larva, they are going to be spreading it hugely. They really are. But that's the case with all of the virus things. You've got these minuscule size particles, and if the bees are doing their stuff and cleaning everything up, what they're actually doing is spreading it. And that's just another picture showing the usual pictures of it there. But it isn't really a tremendous problem. It has been shown to be in the adult hypopharyngeal glands, and of course they're feeding young bees, which shortens the life, and it can speed up the development, and that can lead to poor foraging. But it tends to be fairly self-limiting. I gather the inspectors, they get occasions where it seems to be quite prevalent, and then it disappears again. And of course, there is no treatment. It can survive in the glands of adults over the winter, so it might reappear, but it doesn't ever seem to be a major problem And of course, there's no treatment available. And this is an important one. Anybody doing their modules, please remember, just don't assume that every single virus is associated with Varroa, other than Varroa reducing the strength or the immunity of the colony. It isn't really associated with Varroa. So it isn't a very major problem. But this one's interesting one, chronic bee paralysis, CPPV. Again, mistakenly thought to be associated with Varroa, but no, it's not. It was first reported back in 1933. And here we come back to filterable. It was the standard way by then of finding out, was this a bacteria? No, we filtered it and it's still transmissible, so it's a virus. And he noted down these hairless or black bees, and also that there was an effect of temperature. It's a very strange one, this. And there were loads of discussion in the 60s, the great brother Adam and Leslie Bailey, who I already mentioned, because there's been a lot of dissension about could this have actually been the cause of Isle of Wight disease rather than acarine. I personally find it rather sad that people slag off this man who did all that work on acarine before we ever knew anything about viruses. There are similar characteristics, and I think the most fair-minded thing to say is that quite possibly Isle of Wight disease was caused by possibly more than one pathogen, and Celia Davis puts that in her book as well, and that would be my feeling. If you've ever seen John McMullen's pictures of acarine, you would believe how bad it can be. It's really dreadful. Because we don't see it much now, people tend to, rather dismiss it. It appears in two syndromes. There's type 1, which tends to be the more common, where you get trembling wings and bodies, and bees can be seen. This is where you come to the possibility of mistaken it for acarine. Seen crawling around the entrance, these strange K wings, it means that the wings are held unlinked in a very strange way, in a inability to fly, bloated abdomens, build-up of fluid, and quite often then dysentery, leading to death. There is also a type 2, where you get hairless, black, shiny bees, a broader abdomen, and the affected bees seem to be nibbled by the other ones, which is why they'll end up hairless. And this activity is reduced at a lower temperature, and the poor things aren't allowed into the hive, and then they start to tremble, probably upset. So they also are unable to fly. So you have these two syndromes, and this is from BBase. This is showing the black, shiny, hairless ones. They do look very shiny, don't they? And if you look close, the wings are being held strangely too, and they don't seem to have much hair on them at all. Now I've got to go. I think this might be my video. So, sorry, I'll stick with this because I've got a video that I have to come out of it. You can get both syndromes, deaths more rapid at higher temperature, and some have actually noted the presence of acarine as well. So that leads one to wonder, could acarine act as a vector? Now, it is widespread in a whole load of countries, and it can be present, like an awful lot of virus infections, of course, with no symptoms obvious. And in very severe cases, the pupae can be killed in the later stage. Now, this should enable me to bring on this little tiny video, which comes from Jerry, of, I have to go out, I think, don't I? Sorry, it always takes a minute or two. Right, open. It's very short. Come on. Here we go. Sorry, it takes so long, it's hardly worth it. There we go. We'll play it again.
Do the symptoms come on in the summer, though?
Now, if you watch carefully,
I'm not
you
seeing the video, Pam.
will... Pardon?
I'm just seeing a blue-grey box at the top corner of the screen.
I'm just about to play it. There. Minute. Hang on. I saw it. Oh. Play again. There. Can you see that?
No, you might have to share the screen again onto
Ah.
that extra bit.
Ah. Would I? Right. Oh, yes, you're right. It says your screen sharing is paused. Your screen sharing is paused. I'm clicking on it. It's not doing anything. Oh, how annoying. Let's try down here. Yeah, it just says new share.
That'll be it.
Might that be better? Now, how's that?
Yes, there we go.
Is that working?
Yeah.
Yeah. It's terribly, but I'll play it again. Now, if you play it again, look carefully and you can see the black shiny ones and they're all trembling and the wings are going like mad. This is from Jerry Collins. You can hear them speaking in the background as they're all saying, oh gosh, look at those bees. Can you see them? Sorry, it's very brief, but it certainly is very bizarre. Now, let's hope I can get back to where I was before. Has stopped as the share window is closed. Okay, now I've got to go back to, how do I get back?
Share screen again, I think.
I have, but I've only got up whiteboard. Where's my PowerPoint? Oh, hang on, there.
There we go.
Right, sorry, was that really worth it? I asked myself. Never mind, you did see them trembling, I hope. Now, there's a paper just come out recently from Giles Budge and many others, including David Evans, that you go back, that you're probably familiar with, and Mike Brown. And this came out earlier this year as a serious emerging threat to, I don't like this thing stuck over, that's better, as a serious emerging threat to honeybees. It's a slightly over the top title because they've mapped it around the country. And yes, there is an increase in incidents, but the trouble is, I think you have to be terribly careful how you word your title, because that got picked up out of all proportion by Mail Online. Well, you'd expect it, wouldn't you, the Daily Mail? Honeybees being wiped out by a chronic viral disease. Trembling, he's got this right, trembling, weird, greasy abdomens, inability to fly. But, and the bit at the bottom, honeybees are increasingly suffering at the hands of a deadly viral disease that is sweeping England and Wales. Well, that is desperately over the top. It isn't sweeping England and Wales, but it is increasing. The Guardian was a little more reasonable. Study whether new strain of chronic bee paralysis virus is responsible. Well, I don't think they discovered that it was a new strain, but it is spreading because they've been surveying and they have picked up a lot more. And it's certainly related to, they had to be terribly careful because poor Giles Budge said with larger numbers of colonies, which meant that if he wasn't carefully upset the bee farmers. But I think with all with all the viral diseases, larger numbers of colonies do lead to problems. If you have fewer colonies, I mean, we just talked about sac brood, they're spreading it. So the viruses can only spread if they are able to come in contact with one another. All viruses can only get in by contact. So it is inevitable, really. And there didn't seem to be anything particularly clear about one part of the country, except that it was where there were larger concentrations of colonies. So yes, it is spreading rather, and really all one can do is make sure that you haven't got anything too crowded and possibly re-queen, because with most of these things, the queen may be genetically predisposed to something. Now, those were not associated with Varroa, but we now come to the ones that are associated with Varroa. And here's the little deer sitting on a bee, wretched things. And this is very photogenic, isn't it? But it has, of course, led to huge problems. Before the advent of Varroa, there were very few serious viral infections. Brenda Ball used to joke that it made her career when Varroa came, because whereas before I met her at a microbiology meeting before I'd started beekeeping, and she was bemoaning the fact that nobody was interested in her work because it was only in bees and they weren't very important. And of course, once Varroa came, there were very, very many more. And she worked with Bailey, and they just led the world on the discovery of the links between Varroa and viruses. So some which were rarities have become more prevalent, but not all of them. This is a wonderful example of the joys of a scanning EM. It's a fancy one, it's called a low temperature one, but it just shows the 3D effect, which is wonderful. And it also shows wonderfully the hairs on the bee, doesn't it, as it attaches itself to the surface of the bee. We get acute bee paralysis virus, which is associated with Varroa. Now, this can kill bees rapidly, as the word acute, but without Varroa, you have very few symptoms. It might be there, you'd hardly ever know. The adults spread the virus in the brood food, but this can be a major cause of colony death. And it's linked also with this Israeli acute paralysis virus. They're so similar, these. That was what wondered if it was one of the causes of CCD in the US, or rather contributed to. There are various closely related ones. And then there's slow bee paralysis virus. That affects both larvae and adults. That can appear late in the year. Very few symptoms again were noted. Usually affects front legs. And it was only discovered when they were looking at B virus X, which is equally obscure. It can kill the colony, but they really are pretty rare. It was discovered initially in a UK colony. The distribution's unknown. You see, there's still so little known about a lot of these. The incidence is related closely to the Varroa load. Good reason for us to keep our Varroa load down. And it was hardly seen prior to Varroa. Kashmir bee is another one that's closely related, but it is genetically distinct. The capsid proteins are slightly different. And this came originally from Apis cerana. And it's also been found in a contaminant in preparations of an iridescent virus, which is a rare one from cerana, but nothing we would really worry about. It's found in Australia in mellifera and also in stingless bees. And it's thought to be one of the most virulent. It's a little bit of a misnomer here, I think, in that very few particles are required experimentally, but it has been detected in Varroa, and the distribution seems wide, but it's very low outside Australia. So I've no idea how important that is. This is the biggie, of course, deformed wing virus, closely associated with Varroa, and this is of major importance. It often is the cause of overwintering deaths. When you see a bee with deformed wings, it probably means that the majority of the bees that look okay will be carrying the virus. Brenda Ball used to bang on about this time and again. Just because you only see a handful of bees with deformed wings, don't think the rest of them are okay. They're not. It was first isolated by Brenda Ball in 1983, and it soon became very clear that the deaths were dependent on the load of deformed wing virus. It can be detected in the female varroa mite, but it rarely kills if there's no varroa or very low varroa. I mean, they are pathetic little things, aren't they? And this is a gorgeous little scanning EM of it. They do like to colour them to make them look pretty. And that's from Steve Martin's lovely new BBK special edition. This is a transmission EM from a publication in PLOS One. Don't worry about these, it's something extra they were talking about. And here they are. These are the viral particles. Look how many there are. Masses of them, masses and masses of them. So a nasty thing. Now, Steve Martin's shown that a good sign, the bees appear to be developing natural resistance to varroa. And in the colonies that are doing that, it's no longer causing major damage. I'm not going into detail with that. It is fairly complex. And if you haven't read it, please go and buy one. It's really superb. It was issued just a couple of months ago. You can get it from the BBK online, and he goes into a lot of detail about how to detect whether you've got natural resistance or not. But it is encouraging, I think, that we're beginning to see, if you like, evolution going on. We have some viruses associated with nosema, a pretty dreadful picture of mine, I'm afraid, but here's nosema in the background. Absolutely dreadful. I hadn't got a good link up to my microscope. Nosema, of course, now called a fungus. And what do we have associated with nosema? Well, the most famous is black queen cell virus. Also, filamentous virus. That unusually is a DNA virus. B virus Y. Now, they're not related to one another at all, but they do seem to be associated with nosema. And the link is probably because they all get in via the gut. And nosema, as I'm sure you all know, affects the cytoplasm of the gut epithelium. So it damages the gut and they are able to get in. And it may, they may therefore impair, sorry, the nosema can impair resistance factors. I couldn't find a good picture of black queen cell virus, but the queen cell develops this dark brown black colour and the pupae inside are dead. Early stages, the pupae are pale yellow and it has a tough skin, very similar to sac brood. But strangely, it doesn't seem to multiply much when it's ingested by other bees. So if you do see black queen cell virus, what you've got to do is check your bees very quickly for nosema. There are a number of other rather obscure ones, and to be honest, so little is known about them that I wasn't going to mention them. So where do we go from here? Can we treat viruses? Well, currently, no. Attacking a virus, this goes the same for a human virus, without killing the host cells is difficult. Remember that they've taken over some of our cells and reorganized the cell completely, broken it all down and made it churn out viruses. So trying to attack it is really, really difficult. Developing therapies is very expensive. So nobody is going to develop a therapy, unfortunately, for a bee virus. and it's only likely to come from some other area. So sorry if I've gone on rather long. Thank you very much for your attention. I'll take that off. Shall I stop sharing? So if you have any questions, stop share. Right. If there are any questions that I might be able to answer.
There are one or two. Why does black queen cell virus only affect queens and not seem to affect workers, although it can be found in them?
Don't know. The thing I said at the end, it's just very strange. It's one of the joys of infectious diseases. Who knows? It doesn't seem to do much to other bees. But remember, the queen is very different. She really is. But it's perhaps just as well that it doesn't infect all of them, because the queen there's probably an awful lot. Looks a little bit like a sort of horrible sac brood gone wrong.
I'm just reading through to make sure I've not missed anything. The virus, I can't say that. I really can't say that. The virusy effect on cells sounds like a cancer effect. Is that right?
No, because what cancers do is there are some cancers that are thought to be caused by viruses, but the vast bulk of them, cancer cells, simply enter an over rapid reproduction rate. So instead of reproducing, but they are reproducing normally, whereas here the cell isn't reproducing at all. It's just churning out. It's quite clever, really, isn't it? It's like some sort of terrorist getting in and just taking over the cell and churning out itself.
Right. There's a very good question that I think we'll end on. Are there any beneficial viruses?
Sorry, can you say that again? Didn't hear.
Are there any beneficial viruses?
What a good question. I can't think of any, except the theory of the origins of them are that they were incorporated originally into cells and became part of the cell. But I can't, but that's just probably my ignorance. Because I'm interested in infection, I don't know. I shall have to go away and try and find out, but I certainly haven't heard of any.
Right, okie doke. I think that's all the questions we have time for. We have been online for quite a long time. Thank you very much for your talk, Pam. It's been really, really interesting. It's told us a lot about viruses, not just bee viruses, which has been really interesting. So thank you very much. Thank you everyone for joining us tonight. There are other talks going to be available. If you'd like to keep an eye on the BBK website and our social media for those, then you'll find out about those. We've got a couple lined up coming very shortly. So once again, thank you very much for your talk this evening, Pam. It was excellent.
Thank you. Thank those that joined it. Thank you.
Yes, right. Good night, everyone.